Well, thank you so much. And also thank you for inviting me to participate in this absolutely fantastic week of science. So I want to talk to you about DNA, and I think when most of you think about DNA, you think of it more in terms of the library of the cell. So DNA is this magnificent structure of the double helix, and in that spiral staircase, we have the steps in that staircase, which are the DNA base pairs, and they come in 4 flavors, A, T, G, and C, and that encodes everything that we are. That is our cellular library in each one of our cells. But I want to tell you a different aspect of the chemistry of DNA today, because we think of DNA somewhat differently, and we do so with pictures like this. So this is another picture of the DNA double helix, but I've schematized it for you. We still have those DNA base pairs in the center of this structure. Instead of the sugar phosphate backbone of DNA, that negatively charged backbone, the sides of the spiral staircase, I just have this little ribbon diagram. And that's because I want to focus your attention on these DNA base pairs. But I've made them all the same. They're only coming in one color. And that is because in the context of this chemistry, they are all the same. They're all stacked, one with another, one right next to another. And if you look down the helix axis, you see that they're all overlapping one with another in terms of this stack. Now, the DNA base pairs have this, they're aromatic heterocycles. They have these delocalized electrons that are over all the nuclei in the plane of a given base pair. And so those delocalized electrons of one base pair interact with the delocalized electrons of the next base pair, and that keeps them stacked together, about 2 kilocalories per base step. And it's really a measure, a large aspect of the stability of a DNA helix, of a DNA duplex. Now, there are lots of examples that we know of materials that are similarly stacked. And sheets of graphite, for example, are stacked on top of the next plane, the next sheet of graphite, and that adds a measure of stability. Now, it turns out that those materials that are stacked in that way tend to be conductive. If you pull out an electron from that material, electrons will move through that material and be good conductors. And so essentially, when Watson and Crick came up with the structure of DNA, the same year, it was asked whether or not DNA might also serve as a medium to conduct electrons. And so, since we're not talking about DNA as a material, DNA is not a material, it's in solution, it's an assembly. Instead of talking about the material being conductive, what we talk about is whether or not electrons can migrate, can travel through that DNA base stack. And that was the question. And that was the question that became interesting to my lab. It started, as most experiments do, we thought something else was going on, and we just had to keep on returning to this question. And in my lab, we started looking at this question maybe about 25 years ago, and then labs across the world started looking at it as well. And over that period of time, I won't take you through all the interesting stories. I'll give you a few of the stories. But over that time, what we've learned are 2 important aspects of this chemistry. First, we call it DNA-mediated charge transport. That is, charges are transported through the DNA helix, mediated by the DNA helix. And what we found was that this charge transport can occur over very long molecular distances, and that makes it very unique and powerful chemistry. The other thing that we learned is, although it can go over long molecular distances, it depends exquisitely on the stacking of the base pairs one with another. So anytime you interrupt that stacking, you break the wire. You turn off the charge transport. So let me give you a few examples of that. Here's one example where we took a Rhodium complex. I'm a bioinorganic chemist, and we make these metal complexes all the time. And this metal complex has the nice characteristic that it also has this ligand, this structure that can stack within the DNA helix. It also has these delocalized electrons in that structure. Now, it has another characteristic, and that is that when you shine light on it, it's a potent photooxidant. So it can abstract electrons from DNA. Now, the easiest base to abstract an electron from is a guanine base, and particularly when you have a guanine doublet, two guanines next to each other, that 5' guanine, the left-handed guanine, is the one that's easiest to pull an electron out of. And what we found was if you took this complex and you irradiated it, tethered onto the DNA, that it was able to abstract an electron not just from this guanine, but even from that guanine. And then Megan Nunez in the group made a very long piece of DNA with a lot of double guanines, and she used a different photooxidant that also could stack in the DNA, and she found you could abstract an electron 200 angstroms away from that first photooxidant. So these are very long molecular distances. It allows you to do chemistry then, to do redox chemistry over very long distances and with some measure of control. But again, as I said, it depends on the stacking of the DNA helix. So here's another case where we have one of our tethered molecules. In this case, it's a tethered ethidium. It's an organic molecule that can also stack, intercalate into the DNA via these delocalized pi electrons. And usually, ethidium fluoresces. And it's a good stain, fluorescent stain for DNA. But if you have one of our rhodium intercalators on the other side, you don't see any fluorescence. The fluorescence is gone. It's quenched. It's short-circuited by electron transfer through the DNA. But now, in this assembly, we have DNA of various base pairs, but let's just change one T, a thymine, to a C, so now we have a base mismatch, because T likes to base with A, C likes to base pair with G. But now we have a mismatch, and when we have that mismatch, the fluorescence comes back. It's not short-circuited anymore. And so that's an example where something as simple as a single base mismatch can turn off this chemistry. Now, just to give you some calibration, here are crystal structures of various DNAs, DNA duplexes that are Watson-Crick base paired. And this is fully Watson-Crick base paired. Here's a GA mismatch, and here's a CA mismatch. And I suspect if I hadn't color-coded them, you never would have noticed. This is a very subtle perturbation. Actually, it's more that there's more dynamical motion going on at that mismatch than before. Well, as we were doing these experiments, all sorts of different labs started doing very fancy experiments, single molecule experiments, a lot of physics experiments, publishing lots of papers in different journals, some of which said DNA was a conductor, others said it was an insulator, and everything in between. I'll show you a case where there are 11 orders of magnitude of variation in what people were saying and publishing about DNA. But a lot of those experiments were done at 2 degrees Kelvin in the absence of water. And I don't know what DNA looks like at 2 degrees Kelvin in the absence of water, but I suspect that might have a little effect on the stacking of DNA. And so we wanted to do a single molecule conductivity experiment. And then we saw this paper by Colin Nuckolls and his group at Columbia, because in that paper they were able to measure relative conductivities in water at room temperature. And in this device, what they did was they set up the device, so that they had a carbon nanotube which could conduct, and they would measure the current flow through that carbon nanotube, and then they would use an oxygen plasma to introduce a gap, a small gap, a molecular size gap. And in the center of that gap, because of the oxygen plasma, they had generated carboxylic acids. And so we can now use chemistry where we take a molecule of interest that we functionalize with two amines to make an amide bond in that gap, and then ask, if we flow current through here, what is the relative current flow once we have the molecule of interest versus what it was with the carbon nanotube in the first place. Does that make sense? And you can do that in water at room temperature. Okay, so we did that experiment. I called up Colin Nuckolls, and we took a piece of DNA, and on one end we put an amine so that we could make this amide bond, and on the other strand we also made an amide bond, so we had a nice little robust assembly. Now before we cut, when we had the carbon nanotube in that spot, we had a certain amount of current. We zapped it with the oxygen plasma, and then we had no current after cutting. And then when we put DNA in the gap, we had some intermediate current. So not as much as the carbon nanotube, but clearly there was some current there. And then we did another experiment. We took one strand of DNA and functionalized both ends of that strand. And then we had the other strand, which was not covalently bonded in the assembly. It was hydrogen bonded through the base pairs, and it was stacked together, but it wasn't covalent. And again, what we saw was before cutting, we had a large current, then we had no current, and then when we put in this in the assembly, we had an intermediate current. But if we had this system where only one strand was covalently attached, we could literally wash away that strand. It was just non-covalently associated, so we could peel it away and then float in another strand where we now had generated a single base mismatch. And when you generate that single base mismatch, before, when we had the well-matched strand, we had a lot of current, that intermediate value. But when we now had a single base mismatch, there was no current. Then we diluted that out, introduced another mismatch, a GT mismatch, thermodynamically as stable as an AT base pair, still no current, and then washed that away and added the fully matched complement again, and again we saw an increase in current. And you could cycle this through many times. And that gave us some confidence that we were looking at what we thought we were looking at in this system, in this single molecule array. So, what was the relative conductivity of DNA? Well, as I said, just to give you some sense of things, you could calculate within this molecular size gap, based on the publications that were already in the literature, what the resistance would be across that gap. And as you can see, we've got 11 orders of magnitude variation in what people were predicting in some, I guess, reputable journals. But now you could also calculate if you had graphite within that gap. Now, you could imagine graphite in the parallel direction. There you're going to have very high conductivity. But the analog to DNA is to put the graphite set up so you're looking in the perpendicular direction. And in that case, you would expect, you would expect a resistance of 3 times 10 to the 5 ohms. What did we see? A mega ohm resistance. So the way to think of it is very much like those stacked materials. And if we introduce a mismatch, in that case, what we see is a 2 order of magnitude attenuation in what the current flow is going to be. Okay, so now you can take DNA, you can hook it up onto a gold electrode, you can have a counter electrode in the system, and watch current flow in the system. And what we do is we use an alkane thiol to attach that DNA duplex to the gold surface. And we also introduce a redox probe, a stacked redox probe, at the top of that DNA-modified electrode. and then we apply a potential onto that gold surface, and then we vary the potential until we reach a potential where we have enough driving force to add an electron to that redox probe, and then what you see is you have this blip of current. Okay, these are called cyclic voltammograms. They're sort of funny looking, but they tell you a lot of information about what's going on. Okay, so we have this blip of current, and then electrons are flowing from the gold to the redox probe, and then you can switch the potential, and so now electrons are going to go in the other direction. We're going to see electrons go from the reduced redox probe back to the gold surface, and there's another blip of current, and then you vary the potential and nothing happens. That's with well-matched DNA. Now let's introduce one single base mismatch. Again, we'll change the TA base pair to a CA base pair. And chemists, we can go in the lab and synthesize DNA, so I can synthesize anything I want opposite that A. And when we do that, now you see there is no blip of current. There's no current flow in that system. Now you can actually attach this to an electrocatalytic cycle. You can amplify the signal very much. You can get very large signals. And what you see is an attenuation in the system. If you look at the charge accumulation at the redox probe as a function of time, you can see that there's an attenuation for all of the single base mismatches in DNA, even that GA mismatch, which was so beautifully stacked within the helix. All of them can be detected as an attenuation, irrespective of the sequence context. So just one mismatch is enough to do the job. And in fact, interestingly enough, here are examples of different modified bases. So our bases are constantly getting damaged in our DNA. And these are examples of different lesions, different things that happen to our DNA bases. You can get methylation. You can get oxidation, 8-oxoadenine, just adding an oxygen atom at the end of adenine. And when you do the experiment with one modified base in this DNA duplex, you see this enormous attenuation versus with well-matched DNA. So these small changes in the base, these base lesions, are enough to turn off the electron transfer. So to give you some sense of the experiments that we can now do today, we make these multiplex DNA chips. We can address different DNAs to different spots on the chip. We can monitor protein binding and protein reactions on DNA. Ariel First in the group has come up with a device where we can look at methylation and compare and diagnose tumors versus healthy adjacent tissue based upon how those DNA binding proteins are acting in that system. So it's remarkable how sensitive a probe this is and what a good diagnostic we can develop using that chemistry. And we can even look at very big pieces of DNA. So we did an experiment using 100 mer of DNA on this chip, and we characterized it. And you can distinguish one single base mismatch in the 100 mer just as readily as you could in the 15 mer. And it turns out this is the longest documented molecular wire to date in the literature. It was 34 nanometers of DNA where we could see this charge transfer. So in some respects, yes, it is a molecular wire. I wouldn't wire the house with it. But what it really is a sensor. Because the chemistry is so sensitive to what the lesions are within that DNA duplex. Okay, so we have this chemistry that can go over long molecular distances, that's responsive and reports on the integrity of the DNA, that it reports on proteins that bind to DNA. But this is DNA. This isn't just some molecular copper wire. Does nature use this chemistry? And for the past, I think, 10 years or so, that has now become the main focus of my laboratory. And I wanted you all to get a sense. These are experiments that we're doing all the time. I don't know the answer to all these questions. But that's the exciting thing about doing chemistry. You can go in the lab, design an experiment, and see if it's right. see if it works. And this is sort of a crazy question to ask. But increasingly, what we're finding is that the answer is yes. And that's exciting to think about because this chemistry provides a way to get signaling, long-range signaling, across the genome to respond to events, coordinate events across the genome. Okay, so the first question you have to ask is, well, the DNA in our cells is not just naked DNA. It's wrapped in the chromosome. The DNA is wrapped around histones. This is a structure of the nucleosome core particle in which you see that where you've got the histones and DNA is wrapped around it. And so you might say, well, when it is in this nucleosome structure, the DNA is wrapped. It's not straight. Is it still stacked? Is it still able to carry out the DNA charge transport? Well, the answer is it is. It's much the same. And so if you attach a photooxidant at the end of the DNA and you put it in this nucleosome structure, the pattern of guanine damage that you see is the same whether or not you had the histones there. So we like to think about DNA being packaged, put away for safekeeping, protected in the chromosome structure, but it's not protected from long-range oxidative damage. It's not protected from DNA charge transport in that regard. Now, another question that you need to ask is, rather than just thinking about our little metal complexes injecting holes or electrons, what about proteins? Can proteins, when they're bound to DNA, be oxidized or reduced? And the answer to that is yes as well. And here are two examples from our laboratory. It turns out that pathogenic bacteria, when they infect a host, our immune system responds by generating a lot of peroxide, a lot of oxidative stress to try and kill the DNA, to damage the DNA of the infecting pathogen. Well, what the pathogen does is it generates a large concentration of these proteins, these DPS proteins, that are proteins that have a little peptide around them, but in their inside, they have an iron core, and the iron core was thought to serve to take all that oxidative stress and take those holes, those oxidizing equivalents, and protect the DNA from damage. And Anna Arnold in the group was able to show that that's exactly what can happen. And so if we take a ruthenium oxidant to generate holes in the DNA, we can oxidize the iron in that DPS and protect that DNA from damage. In another example, in E. coli, for example, the E. coli has to respond also to oxidative stress. And it does so using this transcription factor called SOXR. SOXR binds to DNA, and when it gets oxidized, it kinks the DNA in this way, activating that segment of the DNA for transcription, and then a series of genes that respond to oxidative stress are transcribed in response to that oxidation. And so what we were able to show in this case, Paul Lee was able to show that you could, again, take a photooxidant, attach it to the end of the DNA, and activate transcription by oxidizing SOXR. So you can oxidize a DNA-bound protein in that case. So we now know you can oxidize the DNA-bound protein. And so let's think of that oxidation as a signaling route. Does that mean that we can use that long-range oxidation essentially as a means of long-range signaling? And in particular, we thought, well, the place that it would be most advantageous to use this is in the context of repair. We have a system that's sensitive to mismatches, that's sensitive to lesions. Maybe nature utilizes this chemistry for long-range signaling in the context of repair. Now, it turns out that our DNA is getting damaged all the time, especially if you don't eat your broccoli. But when it's getting damaged, it's also getting repaired. And we have this whole machinery of different repair proteins that are constantly finding the damage and fixing it. And it turns out one subset of these proteins are called our base excision repair proteins. Those are finding damaged bases, much like those lesions in the bases that I showed you before. They find those damaged bases, they flip out the base, they excise the base, and then another protein comes and fills in the gap. Now, it turns out some of these proteins go after one lesion. Another protein goes after another lesion. None of them are very specific for the lesion that they go after. Maybe they bind a factor of 100 better than they do to unmodified DNA. And none of those proteins are in high concentration. They're in very low copy numbers, maybe 100 in a cell. And yet, if those proteins don't work, we're in big trouble. In us, with those base excision proteins, if we have a mutation in that base excision repair protein, we'll have a predisposition to colon cancer. So how are those proteins finding that needle in a haystack? Well, it turns out that for a subset, these base excision repair proteins, they have a 4-iron, 4-sulfur cluster in them. They have a little bit of iron. Now, as a bioinorganic chemist, which is what I am, 4-iron, 4-sulfur clusters scream redox chemistry. In our mitochondria, we're filled with 4-iron, 4-sulfur clusters that are shuttling electrons back and forth for oxidative transport. And yet, why on earth would you put this little redox cofactor anywhere near DNA where potentially you could damage it unless you had a reason? Well, when the scientists who first discovered these 4-iron, 4-sulfur clusters in these repair proteins looked, they also said, well, maybe it's doing redox chemistry. So they tried to oxidize and reduce the protein, and they couldn't under physiological conditions. But they were looking in the absence of DNA. And DNA is a polyanion. That's going to change what the potential is, how easy or hard it is to pull an electron out of the protein. And so in my lab, what we did was we took our DNA-modified electrode, and we asked, when the protein is bound to DNA, can you oxidize or reduce it? Could you see a blip of current? So the redox probe is the protein. And indeed, we can see a blip of current, and that blip of current occurs at about 90 millivolts versus NHE, just what you would want for a physiological switch. And it's the same for all of the proteins, the various proteins that we looked at. And then what we did was on a graphite electrode, where we had a bigger window to monitor what was going on, we compared directly what the potential was in the presence of DNA versus in the absence of DNA. And what we saw was there really was a potential shift about 200 millivolts negative. Now, what that means thermodynamically is that the protein in the oxidized form must have 1,000-time-fold higher affinity for DNA than the protein in the reduced form. That's just the thermodynamic cycle from doing this electrochemistry. Okay, so let's forget about the details. Those are the facts. It binds better in the oxidized form. The potential is shifted when it's bound to DNA. So what's our model for how this all works? So here's our model for how DNA charge transport is used by repair proteins. The idea is as follows. You have a protein, and the iron-sulfur cluster is in the 2+ form, and it's not redox active. But now it binds to DNA, and when it binds to DNA, that shifts the potential, so it's easier to oxidize, and it spits out an electron, which it sends to another one of these proteins, a different protein with a different substrate that it's targeted, that's bound along a different site on the DNA. That protein then gets reduced, and the protein then has a lower affinity for DNA, so it falls off, and it goes someplace else. Now, in fact, what I've just described for you is a scan of this region of the genome. The DNA was fine, and so the repair protein went someplace else. But now the repair protein is in the vicinity of a lesion. And when it's in the vicinity of the lesion, it can't do the electron transfer. And so on a slower time scale, it processively moves along the DNA, finds the lesion, and it fixes it. So essentially what I've described is a redistribution of the repair proteins in the vicinity of the lesion. It's a first step. It makes it much more efficient to get in the neighborhood where you need to do the repair. So how do you prove something like that? One experiment that Pam Sons and the group did was to say the following. If I have strands of DNA that are fully matched, well matched, mixed with strands of DNA where we have a mismatch, not a substrate for one of these repair proteins, but something that interferes with electron transfer, the proteins should still redistribute onto the strand where we have a mismatch. And so she made strands of DNA with a mismatch, 3,800 base pair long strands with one mismatch in it. And she mixed it with strands of DNA that were well matched. And then she used atomic force microscopy, where you just tap on the surface where the DNA is, and you can have these beautiful pictures of the DNA, and you can even see where the proteins are. And here's another image. Here's the DNA, and here's a protein bound to it. And then you literally count how many proteins are on the strand that has the mismatch, which we have is a slightly different length than the proteins that are well matched. And what she saw was, in fact, that there were more proteins on the mismatched strand. If both strands were matched, then there was an equal density on the two strands. The binding density ratio was 1. And then what we did was we made various mutations of endo-3, this protein that we were looking at that has an iron-sulfur cluster in it. And what we saw was this direct correlation between how well you redistributed onto the mismatched strand versus what kind of signal you had on our electrode. That is, how good the protein was at doing DNA electron transfer. And what we saw was there was a higher binding density ratio, a higher percent on the mismatched strand if you had a higher electrochemical signal, if you were better at doing electron transfer. And if you couldn't do the electron transfer, then the binding density ratio was 1:1. Couldn't distinguish the mismatched strand from the other strand. And remember, we're talking about one mismatch in 3,800 base pairs. Now, at that point, you might be asking, what distances do you have to do this for this to be of any value? And so we did simple calculations which showed us that actually, if we didn't have a mechanism like this and just thought about proteins sliding along the DNA or looping along the DNA, and we ignored the fact that in our cells, the DNA it's a very crowded situation. There's a lot of traffic, and it's hard to slide along the DNA. If you just do simple calculations, there isn't enough time for these proteins to scan the genome of an E. coli cell just by looping or sliding in the best, most charitable contribution. But if you now have electron transfer events over on the order of a few hundred base pairs, which we were able to see electrochemically, what you see is that you rapidly increase the efficiency, you decrease the time that you need to do this search. Okay, but we also wanted to look and see whether or not this occurs within a cell. And for that experiment, Amy Bohl decided to use genetics. And in this case, what we're looking at is the following. So according to our model, endo-3 binds to DNA, gets oxidized, spits out an electron, and let's say it sends it to another one of these proteins, mut-Y. So mut-Y goes after OXO-GA mismatches. Endo-3 goes after oxidized Ts. So in this process, mut Y then gets reduced, it falls off the strand, and then mut Y finds its lesion. So according to our model, endo 3 helps mut Y find its lesion. They're coordinating their search. They're searching together. So if I knock out genetically, eliminate endo 3, I should see an effect on how well mut Y, how efficiently mut Y conducts its search. And in fact, that's what we were able to see. So in this case, you have a strain where you assay for mut Y's ability to fix mutations. And so if it fixes mutations, you have a low number of revertants. Now, if I knocked out mut Y in this assay, we would see about 100 times increase in the number of mutations. But I'm not going to knock out mut Y. I'm going to knock out endo 3, the partner. And when I knock out endo 3, there's a small but significant effect on mut Y's ability to fix its mutations. Now, you can then add back in a plasmid, a little piece of DNA that encodes endo 3, and when you add that back in, you restore the low mutant count. But you could also add back in a mutant of endo 3, which is enzymatically fine. It can do the endo 3 reaction, it can't do the endo 3 reaction, but it can do electron transfer. Because you want to see, is endo 3 fixing mut Y lesions? Well, it turns out when you add that back in, you also restore the revertant count. So even though endo 3 in this mutation isn't working, it's still able to transfer electrons by electron transfer. But then you look at another mutant. Now let's look at a mutant, Y82A, which is enzymatically active as an endo 3, but it can't do electron transfer. And when you add the plasmid encoding that, you do not restore the low mutant count. So it looks as if they're talking to each other, and they're talking to each other by electron transfer. If you look at Y82A in Pam's AFM assay, it also can't find the mismatched strand. Okay. While we were doing these experiments, more and more proteins were being found to contain iron-sulfur clusters. Here's another one, XPD. It's involved in another repair cycle. This is XPD from archaea. And in each of these structures, what one sees is that it has an iron-sulfur cluster. And it turns out this is an ATP-dependent enzyme. You take this protein, you put it on an electrode, you see the same blip of current. It has a very similar potential. But now when you add ATP, turning on that repair process, what you see is that the signal goes up. The signal depends on the ATP, and the signal correlates with how good it is at carrying out its helicase activity. And so in this case, what we did was ask, well, does XPD also find the mismatched strand? Well, it does find the mismatched strand. If we do the AFM assay, it preferentially goes to the mismatched strand. And if we have a mutant of XPD that can't do electron transfer, it doesn't find the mismatched strand. But now we mixed XPD and endo 3, this protein from archaea and this protein from E. coli. It's just chemistry. They ought to be able to still do electron transfer together. And what we found is that they can signal to one another, that the mixture of XPD and endo will find the mismatched strand, but if either endo or XPD cannot do the electron transfer, they can't do the experiment. Okay, well, Bank's getting up, so I will go a little more quickly, but I want to give you a sense of yet another experiment. We had then another protein in E. coli, which could do another repair process. And what we found was much the same story. You take this protein, you put it on an electrode, it has the same potential. ATP increases the signal. It'll find the mismatched strand. It will help in the mut Y assay. And looking then at the activity of dingy, of this other protein that goes after R loops, we again could see in this assay that it correlates with the ability to do electron transfer. If you can't do DNA electron transfer, there's no growth. The repair protein doesn't work. If you can do electron transfer, in the Y82A case, then you see, if you cannot do electron transfer, then you see there's no growth. We are now looking at more and more and more of these proteins, and it appears, here's yet another one, UVRC, involved in finding lesions associated with UV damage. Again, what we see is that proteins in UVRC, dingy, endomute-Y, are all talking to each other. They're all helping each other by electron transfer among the iron-sulfur clusters. Now, I just want to give you in the last slide a taste of where we're going. We now see that we have this network of proteins that are involved in repair. But more and more of these proteins involved in DNA processing are being found to contain iron-sulfur clusters. Indeed, DNA polymerase in eukaryotes has a 4-iron 4-sulfur cluster in it. Here's a cartoon which represents how our DNA is replicated. It's a very complicated process. We have the leading strand and the lagging strand. Proteins are coming on and off all the time. The process must be coordinated, and you don't want to replicate if there's a lesion in the way. And it turns out that polymerase alpha, delta, epsilon, and primase, the little protein that makes a little primer of RNA to start the DNA synthesis, they all have iron-sulfur clusters in them. And so far, we've looked at primase, and when you take primase and you put it on an electrode, in this case, we're looking just at the subunit with the iron-sulfur cluster, there's no signal. If you do this in an anaerobic chamber, so you can't oxidize it. But now if you electrochemically oxidize it, you bring it to the potentials for oxidation, a signal occurs. You cycle through and reduce it again, and the signal goes away. And you can cycle it through again and again and again and again and again. There's a redox switch, a DNA-mediated redox switch for binding and turning on of primase. And we're seeing similar kinds of things now also with DNA polymerase, Pol delta. So I hope in this story I've given you a different view of DNA. Yes, DNA is certainly the library of the cell. But I think it also, this chemistry, this DNA-mediated charge transport, provides a mechanism for long-range signaling, for long-range communication about the integrity of our genome across the genome. And it's just basic chemistry. There's much more that we need to understand. We're very much at the beginning of this story. But I hope this illustration of how these questions are asked and how these experiments are done gives you some sense of the kinds of chemistry that's being done in my lab and at labs across the world. And so I tried to mention the people that did the work. It's really the graduate students and the postdocs in the lab, and even undergraduates that are involved in these experiments. They do all the work. They're the ones asking the fabulous questions every day. So with that, I thank you very much.
I don't think we have any time for questions now, but I think this is an excellent illustration of discovery and how nature is using what there is. Nature is not reading textbooks in biochemistry. She is using every possibility, and it's just for us, for you, to discover it.
That's
right?
Absolutely.
Thanks, Jackie.
Thank you.